student at the TU Dresden in the Applied Zoology group, and today I would like to talk about sexual selection as a booster for adaptation, a meta-analysis of sex-specific genetic variance in fitness. And the research question we are interested in my PhD project is sexual selection an engine or is it a burden for adaptation? And if you look into nature, you can see all these different kinds of sexually selected traits, these amazing ornaments you can find strong sexual dimorphisms, you can find these crazy beetle penises with spikes on them, and all kinds of ornaments in males. This might make you wonder if sexual selection is not rather costly in the end for the population, because to produce these ornaments seems rather costly. A male combat is likely costly for the population fitness, and these spiky penises, I guess, are not very nice for the female beetles here. So there seems to be a dark side to sexual selection on the population level, but there's also a positive side maybe to it. And one idea here is the Genic capture hypothesis, where the general idea is that male reproductive success is condition dependent, and thereby sexual selection can purge deleterious mutations genome-wide. If you look at the genetic quality of an individual, that likely influences its condition, and thereby condition influences the sexually selected trait you're looking at, and that of course influences the reproductive fitness of these individuals. So through this chain of events, you can have a sexual selection targeting genetic quality genome-wide and purge deleterious mutations. And the idea here is that this works on low demographic costs. That means that if there is stronger sexual selection on males, and reproductive potential of the population is mainly limited by the productivity of females, you could say that in essence, females enjoy the benefits of sharing a gene pool that is purified by stronger selection on males. And I will talk about this point of stronger selection on males a lot today and later in this talk. So Genic capture describes how sexual selection could increase population fitness. And if we look at what data is out there, you find that sexual selection can decrease population fitness. For example, if you look at sexual conflict, one interesting example here, prominent example, comes from Drosophila, where males transfer seminal fluid proteins that harm the female during copulation. But there also can be a bright side with sexual selection increasing population fitness, for example, through Genic capture and purging deleterious alleles. One example comes from these barn swallows, where they monitored a population close to Chernobyl and could show that sexual selection can indeed remove deleterious mutational influx from this population. And there has been a recent meta-analysis showing that sexual selection can actually improve population fitness. So, but all these models of sexual selection, like this Genic capture hypotheses and the models that concern this, and also good genes models, have two major assumptions. The first is that there's a positive genetic correlation between male and female fitness, and that is supported, I would say, by quite a lot of studies today. The second assumption is that sexual selection is stronger in males compared to females, and this would show through, for example, a higher variance in male reproductive success, and that is also known as the first Bateman principle. And this second assumption I want to focus on today. I want to test in our study. So there already has been a relatively recent meta-analysis on this topic, looking at the sex-specific phenotypic variance in reproductive success. So they compiled all these data from these different species, and they looked at the difference between in the phenotypic variance between males and females. And basically what they find is that there is a bias towards males having a larger phenotypic variance in reproductive success. So this is some evidence that actually there's stronger net selection on males compared to females. But what the study does is it relies on the phenotypic gambit. And that basically means that if you go out in nature and you measure a phenotype, and you then draw a conclusion on an unknown underlying genotype, then you rely on this gambit. In this specific example, it means that if you measure the phenotypic variances, you then draw conclusions on the genetic variances that you would expect to find. But this assumes, of course, a correlation between the phenotype and the genotype, or the phenotypic and genetic variance in this case. But this correlation can often be absent. For example, if there's high environmental variance or low heritability, this can remove this correlation. And this is often true for behavioral traits, for life history traits, and for fitness traits. And as we're looking at the fitness traits here, it's important that we check if this phenotypic gambit holds true or not in this case. But why is it important to look at the genetic variance? Why might it be not sufficient to look at the phenotypic variance? One main point here, I think, comes from Fisher's fundamental theorem of natural selection, that states that the rate of increase in fitness of any organism at any time is equal to its genetic variance in fitness at that time. That basically means that if you take the genetic variance, you can look at it as a proxy for the strength of net selection. And that means that the genetic variance basically can be seen as a key component to estimate adaptive potential, and that's what we are interested in here in this question. So we have two main hypotheses. The first is that the phenotypic gambit holds true for this fitness measure, reproductive success. This assumes a correlation of phenotypic and genetic variance. The second hypothesis we have is that the genetic variance for reproductive success is higher in males compared to females. This would show that there's a stronger net selection on males, and this is the assumption of many sexual selection models, including Genic capture. And very briefly to the methods we used, we did a meta-analysis, we searched Web of Science, and we asked for unpublished data on EvolD and ResearchGate. We looked for data on the genetic variance in reproductive success for males and females, so very important that we have in the same experimental design estimates for males and females separately. These are our search terms. I will not bore you to death and read them all to you, they are just here for completeness. And this is what we ended up with. So we have 101 effect sizes in total in 26 different species. You can see that we have some mammal species here, a lot of bird species, one reptile, quite a lot of insects, naturally, with large overrepresentation, of course, of Drosophila species, as it often happens. Then we have one snail species and one flatworm species. And now to the results we have. First, I want to talk about the phenotypic gambit in reproductive success. Just a reminder, this is the assumption that there's a correlation between the phenotype and the genotype, so we can draw conclusions from just measuring the phenotype. And this, here you can see on the x-axis, the coefficient of variation is always the metric that we used, but basically this is the phenotypic variation in reproductive success on the x-axis in males, and the genetic variation in reproductive success in males on the y-axis. And you can see that these two variables are not very correlated. And indeed, if you do the stats, there's no significant correlation between these two. So if you know the phenotype, you cannot directly draw conclusions on the genotype, this tells us. And if we look at the same thing for females, basically the same graph, now phenotypic variance for females here, and on the y, genetic variance for females, you can see that there's also no correlation here. So this highlights that it is even more important for us now to look at the possible differences between males and females in the genetic variance in fitness, and not only at the phenotypic differences. And that's what we did next. First of all, we looked again at the phenotypic variance like this previous study did, and now using our data set. You can see here on the x-axis the phenotypic variance in females, on the y-axis in males. And you can see in orange, you can see monogamous species. That, of course, always is socially monogamous, not strictly sexually or genetically monogamous, of course. And in blue, we have polygamous species. So we analyzed them separately. We used that as a moderator. And you can basically see that most of the orange dots you can see here fall close to this midline, meaning that there's no difference between males and females in the phenotypic variance of reproductive success. Also, I can add that phenotype here basically means that means the number of offspring, and we're looking at the variance of this number of offspring individuals have. But if you look at the in blue at the polygamous species, you can see that most of the data points we have fall above this line, meaning that there's a larger phenotypic variance in reproductive success in males compared to females. This is also significantly so. We used the phylogenetic models to test this. So we have basically replicated the evidence that this previous study has, that at least in polygamous species, we find that there's larger phenotypic variance in reproductive success in males compared to females. And if we now look at the genetic variance, what I think is important, you can see on the x-axis now the genetic variance in females, and on the y, the genetic variance in males. So this is, put very simply now, subtracting the environmental variance that we also include here in this phenotypic variance, and just looking at the underlying genetic architecture and the effect that this has. And again, you can see monogamous and polygamous species separately. You can see that the monogamous species all fall close to this midline, meaning again that there's no difference between males and females in the genetic variance in reproductive success. But for polygamous species, again, most fall above this line, meaning a larger genetic variance in reproductive success in males compared to females, showing that there is a stronger net selection on males compared to females. And this fulfills this assumption of many sexual selection models. So the key messages of my talk are first, that the phenotypic gambit holds not true for reproductive success. We hypothesize that this is due to large environmental variance for reproductive success. And this highlights that it's important to examine the genetic variance in fitness components. And that's what we did next. We looked at the phenotypic and the genetic variance and find that that both of them are larger in males compared to females. But this is only true in polygamous species. We find, at least for polygamous species, a stronger net selection in males. But why is there a difference between polygamous and monogamous species? We would argue this is due to stronger sex-specific sexual selection in polygamous species. And overall, this supports, at least for many species, the assumption of sexual selection models, including Genic capture models, that there is stronger net selection on males. And I want to finish with thanking my colleagues and collaborators. First of all, my PhD supervisor, Tim Janicke from CNRS Montpellier, my co-authors on this study, Maria Maron and Ted Morrow. I want to thank the group at TU Dresden, the Applied Zoology group of Klaus Reinhardt. And I want to thank the numerous people who provided data for this study, because meta-analyses are always only as strong as the primary data out there, and it's always very important that people reply to data requests and such things. And finally, I want to thank the DFG for funding our research, and you for your kind attention. And I think now there's plenty of time for questions and discussion. Thank you very much.